Hi there, and thank you for taking the time to come to my presentation today. My name is Allina Bennett. I'm a master's student at the University of Georgia Tifton campus, where I also work at the molecular diagnostic lab here. Today, I will be talking about the detection of downy mildew pathogen Pseudoperonospora cubensis in spore trap samples from cucurbit fields using advanced molecular techniques. So, brief overview. First, we will look at cucurbit downy mildew, the pathogen. Then I'm going to introduce a novel cucurbit downy mildew spore trap. The methods, results, and ongoing research associated with that as well. So cucurbit downy mildew, otherwise known as CDM, the pathogen and management. Pseudoperonospora cubensis is the causative agent of cucurbit downy mildew. This is an important pathogen to study for several reasons. First, the cucurbit industry is a multi-billion-dollar industry, and P. cubensis is the most devastating pathogen to cucurbits. It is also an obligate oomycete, and therefore it is relatively difficult to study in that we are unable to culture it. Current research efforts rely heavily on field samples as well as spore trapping methods, which I will be talking about today. The pathogen overwinters in southern Florida and moves up the East Coast as the summer season progresses. This has spurned a monitoring system known as the CDM IPM pipeline. This system is able to alert growers if downy mildew is in the area. It is very important to know if downy mildew is going to be around before you see signs of the disease, as that influences chemical control of the disease. Cucurbit downy mildew relies heavily on fungicides, and application of fungicides is most effective when applied before the disease is already established. So what does it look like in the field? On the right, you can see a cucumber leaf that has the classic angular chlorotic lesions of cucurbit downy mildew. On the left, you can see that same plant that is heavily infected with cucurbit downy mildew growing right next to a squash. I find this to be interesting in that they are both cucurbits and therefore presumably hosts for cucurbit downy mildew. Recent research has shown that genetic differences within pathogen populations of cucurbit downy mildew will affect hosts differently. We hope that our efforts here today, what I'm talking about today, are able to contribute to a greater understanding of the pathogen and why it may present like this in the field. So to look at how the disease progresses on the plants, early signs of the disease are shown as water-soaked lesions and therefore difficult to detect. You can see in the middle a leaf, again, with those classic angular chlorotic lesions. That is the disease that has already been established. Once you have severe disease, as you can see in the last image here, the leaves will become completely chlorotic and unable to perform any photosynthesis. You will have any production of cucumbers or whatever cucurbit you're growing stop at this point. So some more traditional diagnostic methods is microscopy. So here you can see what, using a dissecting microscope, different views of Pseudoperonospora cubensis. So on the right here, you can see the black spores on the underside of a leaf. And then on the left, you can see a sporangia on the surface of a cucumber leaf. I find this to be a cool picture in that you can see the hairs of the cucumber here. The sporangia and sporangiophores are in between the hairs, and it demonstrates how this pathogen is able to overcome some of the plant's natural mechanisms to protect itself against things like pathogens. If you go further into microscopy, you can see the characteristic lemon-shaped sporangia of downy mildew, and then on the left, you can see the sporangiophore there. On the right, we at higher magnifications are able to see a flagellated zoospore, which is further confirmation that what you are looking at is downy mildew. While I enjoy microscopy quite a bit, current detection efforts are moving towards a molecular basis, as it is more reliable. Microscopy can require a certain knowledge base and vary person to person. So we are now going to look at the molecular ways to detect downy mildew. In order to increase our collection of downy mildew, we have developed a novel cucurbit downy mildew spore trapping technique, which I would like to introduce now. First, let's look at a commercial spore trap. So this is a rotorod type spore trapping system that is currently being used to monitor cucurbit downy mildew and other similar pathogens. This relies on a rotating axis, as you can see here. A motor will spin this at high speeds where these rods that are coated in grease will be able to pick up whatever is detected in the air. This system relies on a battery, and it is it is a pretty big system and difficult to transport. The homemade spore trapping system, on the other hand, is what I would like to introduce now. This system was developed to be cost effective and use items that you can find in a local store. So here we have a simple D battery powered motor that has a metal rod on its axis that will perform a similar rotorod type style rotation, as you can see in this video here. So the metal rod has attached to it 2 glass slides that are coated in the same grease that those plastic rods on the commercial spore trap would be coated on. This is rotating the slides at the same exact height as the commercial spore trap. It just moves in a slower motion, and we are using the glass slides instead of the plastic rods to collect whatever is in the air at that time, presumably downy mildew spores as well. So to look into a little bit more detail, the comparison between the two, besides the construction, as I kind of mentioned, and the ease of assembly and moving the system from one place to another, cost is one of the biggest differences between these two systems. The commercial spore trap is approximately $1,000 to $3,000. You have to buy it from certain companies, and it comes with a long material list as well as knowledge of how to power something that requires 12 volts of energy in the middle of a field. A homemade spore trap, on the other hand, is only $50. You can use materials that you can find in a local store. It is user friendly. You don't have to have knowledge of batteries to set it up, and it is much easier to transport. So this system sounds great in theory. It uses the same vacuum grease that you are using to collect downy mildew spores with the commercial spore trap, just with a motor that moves slightly slower, and you have a greater surface area with those glass slides instead of the plastic rods. We came up with this in theory, and now we want to make sure that it actually will work to monitor cucurbit downy mildew. So let's get into our methods for molecular disease detection. We placed both the commercial spore trap and the homemade spore trap in 2 locations in southern Georgia. We were then able to perform a modified DNA extraction protocol to both the grease-coated glass slide as well as the grease-coated plastic rod. Once we had that DNA from the extraction, we performed several molecular methods in order to compare these 2 methods and see whether or not it is effective at detecting downy mildew. So let's look at the results. Here we can see that we have the homemade spore trap and some molecular detection methods. So first, we wanted to see if in theory, the extraction from a glass slide using the grease would work using the modified DNA extraction method. You can see that we used fixed spore counts anywhere from 2,000 to 1. PCR was able to detect spore counts from that extraction at 100 spores or greater. qPCR, on the other hand, was 10 times more effective and sensitive compared to PCR, able to measure spores as little as 10. So this means that in theory, using qbensis spores taken from an infected downy mildew leaf, we are able to detect downy mildew spores on this homemade spore trap. We then wanted to see if it compares well to the commercial spore trap and if it is just as effective. Through, again, using a fixed spore count on both the homemade spore trap and the commercial strap, doing that modified DNA extraction, we were able to find no statistically significant difference between the homemade spore trap and commercial spore trap in the qPCR values through those extractions. So that means great, this works in the lab. Now we wanted to see if it does in fact work in the field and you can pick up downy mildew using the homemade spore trap with these methods. So we took samples from the 2021 growing season at 3 different times. The first sample was before downy mildew was found in Georgia, and we were not expecting to see downy mildew through either of these methods. The second one, we were only able to pick it up through qPCR, while PCR did not show any sign of P. cubensis at that time. The last sample, when there was presumably a higher disease load and higher spore count, we were able to detect it using both PCR and qPCR methods. This shows that with a homemade spore trap, you are able to detect downy mildew out in the field. We compared the homemade spore trap to the commercial spore trap using qPCR values and found that, again, there was no significant difference between CQ values of homemade and commercial spore trap when applied in the field. With this, we would like to continue to use our homemade spore trap to contribute to those monitoring efforts. So some ongoing research that we're doing, our collaborators at North Carolina State University at the Casado Ocampo lab were able to develop a disease map using a spore trap in the 2017 growing system. We would like to do the same thing here in Georgia with the 2021 growing season, where gray rods here you can see are where we collected this year, and then this is when Georgia first detected downy mildew. We also would like to use our developed LAMP assay that is specific for P. cubensis. You can see here how it will detect only P. cubensis and not its closely related species, Pseudoperonospora humili. This sensitive technique is very helpful in that we hope one day we can bring it into the field. So LAMP can be applied in the field. You can do a crude extraction. Theoretically, we can do a crude extraction from that homemade spore trap and have real-time knowledge of downy mildew disease pressure. This will influence when fungicides are sprayed. It will influence chemical management of the disease, and hopefully we hope that this will help to combat resistance, which has been an issue in cucurbit downy mildew within recent years. We also hope to do further molecular characterization of cucurbit downy mildew, which again has implications for resistance and pathogen populations going forward. And so with that, I would like to thank our research collaborators, Dr. Allina at North Carolina State University, and Dr. Mary Hosbach, as well as Dr. Julian Bella Rodriguez at Michigan State University. Our research would not be possible without you, and so thank you very much for your collaboration. I would also like to thank our funding agency at the Georgia Vegetables Commodity Commission and my fellow lab members. Thank you for coming to my presentation today, and I'll be happy to answer any questions.